Over. पूरे लूज कनेक्शन है क्या? Let's start. Hello, welcome back to Botany second year students. So I hope that you have done with the complete preparation by today, isn't it? Yesterday only we have finished all the SAQs and the LAQ part. That is, one LAQ is sufficient, whatever you have to learn. So that is what we did with the sewage treatment and the plant breeding. Plant breeding, tissue culture, and sewage treatment. These are the 3 LAQs. आ रहा कि नहीं? देखेंगे. We are now going with the second year botany class. That is till now we have finished the SAQs and the LAQ part from different chapters. And the last chapter that was left out with the FAQs is the 6th chapter, that is the growth hormones. So in this growth hormones, yesterday we have seen the auxins, gibberellins. Now the rest of the ones are the cytokinins, ethylene, and the seed dormancy. Okay, cytokinins. Explain a brief note, write a brief note on cytokinins. Okay, and in otherwise explain the horticultural usage of the cytokinins. Anything they can ask, whatever may be the cytokinins, we have to say. So cytokinins are the growth promoting hormones, which are phytohormones. Phytohormones means they are made in different parts somewhere else in the plant body and stored somewhere else in the plant body. So those are the phytohormones. So the cytokinins are the growth promoting and the phytohormones, and these are going to be synthesized more by the matured leaves and the matured fruits and stored in the buds and in a small quantity in the roots also. These are the cytokinins. And these cytokinins, what they will do, what are the physiological effects of cytokinins in the plant body? In the plant body, cytokinins will do, the first one is the internodal length elongation. Okay, otherwise cell elongation we can say. Cell elongation. In the cell elongation, what we have to say, these cytokinins will enhance the cell division. What they will, we have to write here, we have to say that they enhances the cell division and so that more number of cells are formed and the growth can be seen for the plant body. That is the cell elongation. So which makes the cells to be elongated more. That is the cell elongation. Next one is increasing the, enhances the parthenocarpy. Enhances parthenocarpy. Enhances parthenocarpy means it encourages, otherwise it makes the plants to go with the parthenocarpy. Okay, example, apples and the tomatoes. And the third point, along with the auxins, it is used in the tissue culture. Okay, along with auxins, it is used in the tissue culture. How it is going to be used? Just like the auxins only. In tissue culture, we will use this. In tissue culture, we will use the cytokinins and auxins in different ratios. Okay, when auxins are more and cytokinins are less, then they will promote the root formation. And when the auxins are less and cytokinins are more, which makes the stem elongation. Otherwise, stem formation. Okay, we cannot say elongation. We will say it as stem formation. Okay, so like this, we will use along with auxins, cytokinins in the tissue culture lab for growing the tissues and the otherwise a cell, which is helpful in the biotechnology. And the next one is it controls the apical dominance. It controls the apical dominance. That means whatever the auxins are doing, it will stop that activity and makes the reversible actions in the plant body. That is it controls the apical, it stops the apical dominance. Stops the apical dominance. Okay, that means what it will do? It will make the to stop the auxin activity and makes the branch formation more. Makes the branch formation more in the plant body, because auxins will stop the branching in the stem, but this cytokinins will enhances the branching. So that's the one what we are saying over here. Okay, it enhances the branch formation by controlling the auxin activity, and it promotes the lateral bud formation also more by the activity of the cytokinins. And the next point, what is the next usages of this cytokinins? Cytokinins will help out in the lateral bud formation. Okay, so like that we can say, and even just like the auxins, cytokinins are useful for differentiating the sexes in the plant body. That means the male flowers can be converted into the female flowers by the cytokinins, just like auxins. And these are the most important physiological activities of the cytokinins on the plant body. And now comes the ethylene. What are the physiological activities of ethylene? Physiological activities of ethylene. Ethylene is a growth promoting hormone, otherwise growth suppressive hormone. It's a growth inhibitor, growth suppressive hormone. Ethylene is a growth inhibitor. Ethylene is a growth inhibitor which is present always in the gaseous form. Always in the gaseous form. Okay, so what it will do? The most important activity of the ethylene is it is also a phytohormone. Okay, it is this is present in the gaseous form, and it is also a phytohormone. Okay, and where it is going to be synthesized? It is synthesized more in the matured fruits, young fruits, and stored in the matured fruits. Okay, and this is the most important one. So what are the physiological activities? First of all, this helps out in the ripening. It hastens the ripening process in the plant body. That means it makes the ripening fast in the plant body. This is the first one. Okay, every plant we can use it for the ripening as a process which has to be occurred very fast in the plant body. And the next one is it is helpful in increasing the shelf life and the vase life of the It is used in increasing the shelf life. Shelf life means it increases the leafy vegetable life in the cold storage. Okay, we can preserve the leafy vegetables without it going with the that is loosening its life and the healthiness in them. That is the shelf life. That means we can store the leafy vegetables for a long period in the cold storages, in the refrigerator. That is the shelf life. So example, spinach leaves. Okay, or else coriander leaves. Anything we can give as an example over here. And the next one is was life. Increase the was life. Was life means the flower life. After plucking the flowers from the parent plant, it has to show the liveliness for making the bouquets, isn't it? So here was life is that. Okay, so here we can increase the flower life. Flower life is increased in flower vase. That is the ethylene only can do. Okay, and next one. It decreases the, otherwise we can say it stops the rate of transpiration. Okay, it reduces rate of transpiration in the plant body. So once the transpiration rate is less, means growth is less. That is the growth inhibitor what we are saying for the ethylene. Same thing we are writing in the point also. So what it will do? It reduces the rate of transpiration from the plant body. Okay. So next one. It reduces the cell division. Okay. The cell division is going to be reduced. Cell division is reduced by using ethylene. By using ethylene. Okay. And next one, it is used as the, in some plants, it will be making the parthenocarpy to occur, and in some plants, it suppresses the parthenocarpy. Okay, like that, we can make this ethylene usages in the agriculture and in the horticulture. So the first one is the triple action. Otherwise, it makes the fruits to be ripening. First of all, the fruits ripening is useful in the horticulture for getting the more ripened fruits in a short period of time. And the next one is it controls the apical dominance, and it makes the cells to stop the division itself. Cell division is going to be reduced by the ethylene. So the growth is going to be stopped for a period of time. Okay, and the seed dormancy can be increased in some plants. In some plants, seed dormancy is increased. Okay, and it shows the shelf life and the vase life. That means it will make the leafy vegetables to be stored very for a long period of time in the shelves and in the vase life. That means the flowers can have the liveliness in the flower vase for a long period of time. That is only done by the ethylene. Okay, so like this, and we can, and it reduces the rate of transpiration in the plant body, which is the most important one. What we can say, it decreases the growth rate in the plant body. So it is a growth suppressor, otherwise the growth inhibitor hormone. Okay, and the next one is the seed dormancy. What is meant by seed dormancy? The resting period. Dormancy means resting period for the seed. Dormancy means resting period. So how can we say this? When the seeds are not starting their development in the suitable conditions also, that is called as the seed dormancy. And this seed dormancy can be increased by the some hormones. Otherwise, naturally, some seeds are having the dormancy, much more dormancy period in them. This occurs by the presence of the thick seed coat. Thick seed coat. By the presence of the thick seed coat, it increases the seed dormancy and it decreases the germination point of view in the plant body. And this seed coat is showing the impermeable to the water. Impermeable to water. That is, whenever the thick seed coat is there, it cannot absorb the water. It cannot absorb the water to start its further germination. That is the impermeable to water. It will not absorb and makes the water to go into the cotyledons. So where the seed is not ready to start its germination. And this is the naturally occurring, and the temperature and the water availability also very, very important. If the most suitable temperature is present, then the seeds will stop the seed dormancy in them. That means the seed dormancy can be decreased with the suitable presence of the temperature, and they will start their germination. They will start their germination. And if the soil molecules are having the good capacity of storing the water as well as the other important elements in them, then also dormancy period can be reduced and it makes the seeds to be permeable. Okay, and by the hormones also, that means by the abscisic acid. If the abscisic acid is more in the seeds, then the seeds will be in the dormant stage only. That is, the resting period will be continued more by the growth inhibiting hormone. That is, the example is the abscisic acid, which makes the seeds to be more dormancy periods can be seen in the seeds. That is the abscisic acid. This is a growth inhibitor. And in some plants, we'll see the ethylene also makes the seed dormancy. Okay, which increases the seed dormancy. That is in some plants by the ethylene. So like this, we have to write the seed dormancy, and we have to end up this part. So with this, we have finished all the important SAQs from all the chapters. Now only we have to go with the 11th chapter, that is rDNA technology. the process of rDNA technology and the tools for rDNA technology. These are the two electives left over for us, where we have to go if we have written a little bit in the theory, that is in the IP examinations point of view. For these two questions also, at least we can score 4 marks or 4.5 marks out of 8 marks. So that will be also a good one for us. Okay, so here we will go with the first one is the process of rDNA technology. Process of rDNA technology. So in this, what we have to write? The rDNA, what is meant by rDNA? Then we have to say how this process is going to be continued. The rDNA means recombinant DNA technology. Okay. Recombinant DNA technology. rDNA means recombinant DNA technology. What we have to say this, the definition for the recombinant DNA technology. This one will cut short, say as rDNA. When a new DNA was made by inserting a small fragment of the desired DNA, desired gene, to change the complete organism genotype is called as the recombinant DNA technology. For this process, we have to follow the different steps. That is the 7 different steps need to be done over here. So the first one is the extraction of the desired DNA. Selection of desired DNA. And after this, what we have to take, we have to collect the DNA. Collect the DNA from the organisms. Okay, so first of all, what we have to take, we have to collect the DNA from the organisms. When we are doing this, we have to take out the cell, that is the organism through which we are collecting. Selection of the organisms for the collection of DNA. So in this, what we have to select? We have to select the cell which is having our somewhat desired characters. Otherwise, the DNA where we have to go with the rDNA process. And then that cell has to be, the cell wall and the plasma membrane need to be separated. That means removed so that the genetic material can be taken out separately. So here, first of all, we have to, what we have to do? We have to select the organisms where we have to separate their DNA from them. Okay, that is the selection and the collection of genetic material. Selection of the organisms. Okay, so in this, we will take out the organism cell, and in that the cell wall and the plasma membrane. Cell wall and plasma membrane. If these are present, all these has to be removed first. Okay. So once this was treated with the specific enzymes, cell wall will be digested. Then once only the plasma membrane is present, then this cell will be dipped in the somewhat concentrated soap water so that the microorganisms and the plasma membrane can be disintegrated, so that the cytoplasm and the genetic material that is present in it will be taken out. Okay, it can be separated very easily. That is the one what we have to do in the first step. So here, when we are taking out the cell, then the cell may be the plant cell. When the plant cell we have taken, then we have to use for the digestion of the cell wall, we have to use the cellulase enzyme. Okay, when we have taken the fungal cell, then the chitinase enzyme we have to take to remove the cell wall and the plasma membrane to separate the genetic material and the cytoplasm from them. And if we have taken the animal cell, then it is having only the plasma membrane, no cell wall for it. So it can be, we have to use not the animal cell here, wait, bacterial cell. If a bacterial cell we have taken, then it will be having the cell wall. Bacterial cell is having the cell wall. Then the cell wall can be separated by using the enzyme ligase, not the ligase, sorry. By using the enzyme cellulase, so that the bacterial cell wall can be degenerated very fast, and only the plasma membrane and the cytoplasm can be separated. And after this, we have to take now we have to use the centrifugation, otherwise the separation with the filters, so that we can separate the genetic material. Now, the second step is separation of genetic material. So in this, what we will see, we have to separate the genetic material from the cytoplasm through the filtration. So once we have taken out removed the plasma membrane and the cell wall, immediately the cytoplasm along with all the cell organelles will be taken out in the test tube, and that has to be gone with the centrifugation. We have to do the centrifugation process so that all the cell organelles are disintegrated, and the genetic material, which is heavy, will be collected at the bottom of the test tube. Okay, so like that, we will see once in the test tube, we have collected the genetic material at the bottom, then it can be filtered very easily. And now, how to take out this genetic material? This genetic material can be removed with the elution method, that is with the copper wire. We can easily put, and some fragments will be easily floating in the cytoplasm. So this can be removed with the copper wire, through which we can easily separate these floating fragments of the DNA, so that it is easy for us to go with the rDNA technology. Now, in the third step, what we have to do, we have to cut the DNA at a specific site by using the restriction enzymes. So here, when we have taken out the genetic material from the organism, that is, when we have taken the genetic material from the organism, means here the host and the desired DNA, both we have taken out at the same time, simultaneously. Okay, in two different petri dishes, in two different test tubes, we did this experiment, and now it's the genetic material is ready with us. Now this has to be cutted. Now the third step is cut the DNA at specific locations. So for this process, we will use the restriction enzymes. We'll use the restriction enzymes, and these restriction enzymes will cut the DNA at the specific locations. And these restriction enzymes are again of the two types, restriction endonucleases and the restriction exonucleases. Restriction endonucleases and restriction exonucleases. Restriction endonucleases means they will cut the DNA strand in the center. At a specific locations. Whereas if we take the exonucleases, they will cut the DNA strand at their ends. That is the restriction exonucleases. Like this, we will use the different restriction enzymes for the different purposes. That means where we have to cut the DNA with the restriction enzymes, so that we can use that type of exonucleases, otherwise the endonucleases. And then these restriction endonucleases, these restriction enzymes cut the DNA into two different ways. That is the sticky end and the blunt end. Sticky end and blunt end. Blunt end means when the DNA strand was cutted, both the strands are cutted at the same location, so that it leads the straight cut for the DNA. This is called as the blunt end. And sticky ends. Sticky ends means when the DNA strand was cutted at two different locations for two strands, so that it will give one strand short arm and another one is the long, and one arm long and one arm short, like this. So uneven cuts can be seen for the two strands, so that the two strands can be joined again very easily. That is the sticky ends, what we will say. Like this, mostly we will use in the rDNA process only the sticky ends. So the restriction enzymes which will make the DNA as the sticky ends are the EcoR1. The restriction enzyme here, what we are using is the EcoR1, which makes the DNA cuts the DNA at the specific location. That is a palindromic. It recognizes some specific palindromic sequences, so that it can cut between G and A. So what is the palindrome it can recognize? It can recognize a 6 nucleotide palindrome as GAA-TTC. This is from the 5' to 3' end. Okay, and it cuts the palindrome at the in between the G and A, so that the strands will have the sticky ends for them. Like this, we will cut our desired DNA as well as the host DNA, where we have to make the DNA as the recombinant DNA. Next comes with the next step is, after cutting, we have to separate the strands, isn't it? For separating the strands, we have to use the gel electrophoresis machine. Separation of the specific separation of the DNA strands. Now the step is separation of the DNA strands. Not the strands, DNA fragments. Okay, how to separate these DNA fragments? DNA fragments can be separated by using the gel electrophoresis machine. So what it will do, this gel electrophoresis machine is having an agarose gel in it. Agarose gel in it, which is having the stain also. So that is the ethidium bromide. Ethidium bromide. This stain is also there. Once we are keeping we have taken the agarose gel, in that we kept this ethidium bromide staining also, so which gives the color for the DNA strands according to their length. Otherwise, what we can say, according to their molecular weight. So if the DNA fragment is much more lengthy, means the weight will be more molecular weight. According to that, it will go with the giving the dark color, otherwise the light color for the DNA fragments. So once we kept this, we have taken this agarose gel electrophoresis machine and kept the complete solution in that. Then once we pass the current according this, that is, it is having the 2 electrodes in them. Okay, 2 copper electrodes, and the bigger fragments are collected at the near the electrodes, and the smaller fragments are collected far away from the electrodes, so that the all the different size of fragments will be separated, and the different locations they are going to be separated. So based on our interest, now we can take out the fragment and we can go with the further steps in the rDNA technology. And this ethidium bromide, what it will do for the fragments which are having the high molecular weight, it imparts the dark orange color for it when we are observing under the high power electron microscope. When the light stain was there, that means the light orange color is there, means the molecular weight is less for the fragments. Otherwise, those are the small fragments. Okay, so that they will get only the light orange color bands on the agarose gel machine. So like this, whenever we have seen the light colored and the dark colored orange band, that means the fragments were separated properly, so that we can easily take out the slots and we can go with the next further steps in the rDNA process. And after this, what we have to do after the separation, we have to make the prepare the recombinant DNA. Okay, so how to do this? We have to join the desired gene now to the DNA fragments which were separated by the gel electrophoresis to make the DNA as the recombinant DNA. Otherwise, recombinant DNA. Okay, so in this one, by using we can join the DNA fragments by using the enzyme called as ligase. The enzyme that is used here is the ligase enzyme. Okay, so with this we can make the rDNA is ready for our observation. After this, the next one is amplification process. Amplification means making the multiple copies of the rDNA in a less time period. That is called as the amplification process, otherwise polymerization of rDNA. Anything we can save over here. amplification. Amplification means making the multiple copies of the rDNA which has been made by this process in a less time period is called as the amplification. For this one, we will use the technology as the PCR technology, polymerization chain reaction. Okay, so through this technology, we can make the 1 billion copies in a less time period of the one recombinant DNA. So like that, we can go with the research work very easily, whether all the characters are exhibited in the organism or not with the rDNA technology. So for this one, we need the 2 sets of primers. Two sets of primers were needed for this, as well as we need the DNA polymerase enzyme. Okay, that also Taq polymerase enzyme, DNA polymerase enzyme of the Taq polymerase enzyme. This is very, very important. Okay, so whenever we have used these things, then we can make the multiple copies very fast. Okay, so with these 2 primers, we can add up . So first of all, we have to supply the heat energy so that the DNA 2 strands are going to be separated by the supply of heat. Then to this 2 strands, now we can add the primers. These primers should be added only at the 3' end of the DNA strand, so that the primers will keep on continuing in elongating the new strand in the 3' end. Okay, so like that, and here which enzyme we have to use? A Taq polymerase, so that this polymerase enzyme will be active in the high heat temperature also, and like that we will get the new strand. Like this, many times this process is going to be repeated, so that in a less period of time we will get the 1 billion copies in 20 cycles itself. Okay, so like that we will make the PCR technology to be useful in the rDNA technology, rDNA process. After that, we have to make the after the PCR technology, we have to insert the rDNA into the host. Insertion of rDNA into vector. First of all, into vector. So through the vector, we can insert this. So once we have done with the vector, so the vector will be the one which will make the easy passage of this rDNA into the host cell. And through in the vector, once we have inserted, that will become the much more easiest work for us. After that, we have to insert the rDNA into the host cell. Okay, then in this one, we have to take out the different methods for insertion of with the help of the micro injections, otherwise with the help of the elements. Okay, otherwise with the help of the bacterial plasmid DNA, otherwise the fungal DNA, otherwise the viral DNA also, we can take it for insertion of the rDNA into the host cell. Like that, we can insert the host cell in different methods, and then we have to see whether this DNA is working properly or not in the host cell. That is what we have to say is the downstream process. After checking the DNA is working properly in the host cell, that is producing the enzymes properly in the host cell or not, then we have to commercialize. That means we have to release this product into the outside environment. That means into the for the commercialization process, so that everyone can use this rDNA for their other purposes in different organisms. Like that, we have to do this process of the rDNA technology process. Okay, and by this we have completed for today's session, and we will meet at the same time in the same channel for the second LAQ from this chapter. That is the tools for the rDNA technology, and stay tuned to the same channel. Right?